Okay, so today we are going to talk on 16S RNA, rRNA sequencing and ribosomal database project. Again, this is the part of the same topic, that is microbial diversity and classification, systematic classification. So this is like ultimate where we can categorize the species in different hierarchies from different belonging to different hierarchies based on the characters, phenotypic and genotypic characters. Now, what is 16S rRNA? We already have discussed in my earlier lectures in detail, but today we'll talk more on the database projects and that involve in the sequencing data of ribosomal RNA of different microorganisms, where this database can be used for the prediction of or for the analysis of the different sequences which has been identified from unknown microorganisms. Okay, now we start 16S rRNA sequencing and ribosomal database project. This is the part of the same microbial diversity and systematic classification, but in this topic we discuss about the whole database of the large number of sequences which are generated from different microorganisms belonging to different group, class and orders and phyla, and of different ecological habitat. Now, just to introduce about the advantage of this database is that this database can help us to pick up the sequences quickly and identify the organisms belong to which category or which species. So these are some content and learning points where we talk about what are ribosomes and what is ribosomal RNA, significance of 16S ribosomal RNA and ribosomal database project, right? And then comparison with latest databases which are available, and application and limitations of ribosomal database project. First we should learn what the ribosome is. Now, ribosome is basically a Greek word originated from Greek called ribos, that is ribonucleic acid, and the soma is a body. So it is a membrane-bound organelle ribosome which is present in the cell, demarcated in eukaryotic cells, where the protein synthesis takes place through the translational process. What are the different steps involved in the synthesis of protein? Basically, it's double-stranded DNA is formed after replication to transcribe into the RNA, that is mRNA, and then from mRNA it bound to the ribosomes and form protein synthesis, protein formation. Now, this process of whole protein synthesis as a part of central dogma, as you are seeing here, this is the cell and this is the DNA present in it, and this is how the messenger RNA is coming out from the cell, and this is the ribosome. Now, this ribosome is composed of small subunit and large subunit of ribosomes, both in prokaryotes as well as in eukaryotes. The system is applicable, and then it forms the protein synthesis. This is how the amino acids are synthesizing and protein synthesis take place. Now, this is the whole process of the protein synthesis through ribosomes. Now, what is the significance of this ribosome, and what is the ribosomal RNA? Ribosomal RNA is basically non-coding sequences, right? It from structures and functional core of ribosome. So ribosome is basically composed of RNA and protein, right? In prokaryotes, the ribosomes has two subunits, 30S subunit we call a small subunit, and 50S subunit we call the large subunit. So this is the large subunit, this is the small subunit, this is the small subunit, this is the large subunit. Similarly, in the eukaryotes, we have 60S ribosome which we call the large, and 40S ribosome we call the small subunit. So these are the composed of, so these are the two different components, small subunit and large subunit, and then it form RNA trapped in these ribosomes, and these ribosomes can form the protein through this mRNA by decoding. Now, in prokaryotes, the ribosomes has two subunits, 30S and 50S. In eukaryotes, you have 40S and 60S. Now, different ribosomal RNA, or we call rRNA, are part of these subunits, like 16S rRNA, 30S subunit, then 23S rRNA, and 5S rRNA is the part of the 50S ribosome. So, these ribosomal RNA, which is the part of the ribosomes, they are again categorized in terms of like 16S, 23S, and 5S rRNA. what is so important about the 16S ribosomal RNA? This we try to understand. So the whole lecture, or the whole classification rather, so far we have discussed in last at least couple of 4 or 5 lectures, everything is based on this concept which need to be understood. What is the 16S ribosomal RNA and what is its significance and why it is so important? So the specific type of ribosomal RNA about 1500 nucleotide longs, so almost 1.5 kb, which is standard size, found only in the small 30S ribosomes in case of prokaryotes, subunit of prokaryotic ribosomes, and highly conserved and doesn't change much across the evolution. So this is the most important point, third point. So we should understand what does it mean. What does the conservation of these sequences means? Why these sequences are conserved, and what is the advantage of these sequences to be conserved? So highly conserved sequences which doesn't change much across the evolution. Over the period of time, they are not changed. And that is the beauty of the 16S ribosomal RNA, or 16S RNA, to look into and to check and to analyze about the species identification. It is variable regions that allow us to distinguish different species. So there are two things. One is it is a conserved, and then it is a variable region within the species of that domain, so that we can identify it based on these variable regions compared to the other ribosome of the other species, right? Widely used in bacterial taxonomy, phylogeny, and identification of the bacterial species. Now, if you look at the secondary structure of the 16S ribosomal RNA, right, from Escherichia coli, secondary structure of 16S ribosomal RNA from Escherichia coli. So this is the sequence, right? So this is like A, T, A, G, T, 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 and so on. So this is how the sequence run, and that give you 1.5 kb or 1 kb or 1.2 kb, whatever, depending upon the species. And this is the structure of the, no, you just looking into the secondary structures, you're looking into the hybrids of the RNA, but usually if you ask the definition of a RNA, it is a single stranded nucleic acid molecule. Now, though RNA is a single stranded nucleic acid molecule as mRNA, but there are some certain RNA molecule which are also double stranded RNA molecule. In case of this secondary structure RNA molecule, we are looking at the loops like structures, right? So this is the single stranded RNA, this is the double stranded RNA, see here. Now these loops formation takes place, we call the secondary structure of RNA, and these RNA has some catalytic activity. We'll talk later what does the catalytic activity means. Now if you look into the conserved region of this RNA, you see the variable regions like V1, V2, V3, 4, 5, right? So when you PCR used to amplify these V5, V6, V7, right, or we can have certain other regions which are conserved regions as a PCR amplicon, right? And then we compare with the other species, right? So suppose conserved regions, these are all conserved regions, 1, 2, 3, 4, 5, 6, 7. These are conserved regions, and these are the hyper variable regions, V1 to V2, then V3, V4, 5, 7, and V7, 9. Now, what does the hyper variable region means? So basically conserved regions identical across all bacteria and archaea and use the universal primer. So the beauty of this 16S RNA, 16S rRNA is that the universal primer can be used. So once you identify it, once you check the primer on either side, and these primers are conserved primers, and these primers are conserved primers, so you need not to worry about the making the primers of each species. For example, you have thousands of different species, right, different genera, and you want to get the 16S RNA of different species, so you need to have like 1,000 primer. No, you can only synthesize a single set of primers because these are the highly conserved sequences. That's the beauty of, try to understand, these are the beauty of the 16S RNA conserved sequences. Now, where is the variation? The next question, how this same primer can amplify the sequences, and the sequences vary from E. coli, from Klebsiella, from blah, 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 blah. This is the questions. So, this is because of these variable lesions. See, the red one, green one, blue and violet. So, these variable lesions give you the clear-cut picture about the variation among the species. Clear? I am sure it is clear now. So, hypervariable lesions which are present in that, which differentiate between the species to species, genus to genus, and it discriminates between the closely related bacteria. For example, there are 9 hypervaluable lesions I have just talked about it, V1 to V9, and some regions are better at distinguishing certain taxa. For example, V3 to V4. So, V3 to V4, the blue one, is most commonly sequenced for the microbiome studies, where V1 to V2, that is a red sequences, red region, is basically good for differentiating certain gram-positive bacteria. So, this information which we identified in the gene through this gene analysis or gene sequencing through primer will be used for the analysis of these species. Now, then come to the, I think now you should be clear what the Ribosome Database Project means. Now, Ribosome Database Project is a repository aligned and annotated bacterial and archaeal 16S rRNA sequences and fungal 28S rRNA sequencing. Now, in Ribosomal Database repository or Ribosome Database Project, we have to, the scientists or the group of different scientists or teams of scientists, they identified these sequences and annotated these sequences, and then they form a whole database. Now, the vision of Carl Woese, if you remember, the Carl Woese who gave the concept of domain, 3 archaea, bacteria and eukarya. The Carl Woese and his colleagues, Gray Olson, later worked together on this project. And RDP, that is Ribosome Database Project 1, first version of RDP released on 5th January 1992. So, this is the historical part, hosted by the Argonne National Laboratory. So, this is the history background about this database. Now, the second version in 1995 onward moved to Michigan State University, where it has been maintained since now. Now, we are maintaining, scientists are maintaining this in the Michigan State University. The latest Ribosomal Database Project include the analysis tools, phylogenetic tree and screening services. So, this project started with the storing of or the aligning the sequences of different species. For example, if you have a species of like, for example, Cetacea lepagi, which is very rare organisms in clinical setting, and Cetacea lepagi sequences may not be present in it. So, if you got some new sequence which is not aligned at all in the database, it means it must be belong to certain new. Then you have to think what this new species is. So, once you identified that species, what the species is, then you can easily compare that as a new, put this database as a new species and give the new name for that species. So, this is how the database works, the database advancement version works. How are the bacterial species identified using the ribosomal database? Ribosomal RNA sequence obtained from the International Nucleotide Sequencing databases such as the GeneBank, EMBL, DDBJ, then 16S rRNA from an unknown bacterium, unknown 16S rRNA research against the ribosomal databases. Then we match with the 16S rRNA E. coli, hence unknown bacterial identification can take place. So, this is how it works, and using the sequencing database, using the 16S rRNA sequencing database, you can easily identify these different bacteria. Now, one thing is which I would like to explain you about this database is that better is the database or more comprehensive is the database, the availability of all possible sequences is the database is carrying, then only you can use different tools to analyze the database. Now, if you go back to the same slide, I've talked about the addition of the analysis tool, that is the phylogenetic tree, screening, services, analysis, sequence analysis. So now that in latest database, they have added these features where you can analyze the available sequences, you can get these sequences done to know what the sequences belong to, which species and which genera. So these are all updated version of the databases, and this is how it works. You get it. Now, this is just a comparative chart, an updated chart for this Ribosomal Database Project. So if you compare the 1992, first version, first curated ribosomal database is in 2,700 rDNA or 16S RNA sequences, right? In 1995, when it moved to the Michigan State University, we called RDP2, extended to bacterial and archaeal 16S RNA and 23S RNA, fungal, LSU, web-based tools, aligned probes, match trees, and builders. Now, in 2007, this Ribosomal Database Project classifier introducers, we introduced that into the new Bayesian classification system, rapid taxonomy, assignment widely adopted in microbiome studies. Now, in 2010, the database is basically, in 2010, database tool expansion takes place and large scale sequences inclusion NGS, that is new sequencing platform era where the whole genome sequencing was done. In 2020, training sheet number 18, so we are giving the update features and we call it improved genus coverage, curated high quality sequences. So once the NGS is introduced, that is whole genome sequencing is introduced by Illumina and by other platform, then the refinement of the data takes place, and this refinement is totally based on the genome data. And now to 23 and onward, the taxonomy, more than 600 genera and more than 2,500 species have been added, updated taxonomy, new phyla names, and then have GTDP alignment. Then we have another version, 2.14 version, where we have optimized the training sheet 19, that is the earlier one, improved the speedy accuracy. So now speedy accuracy means like once you add the data sequence in the data, the sequences, you try to fetch it out which sequence it belongs to, and that the process is very fast. So the database project, Ribosomal Database Project is keep updating, and they have got now the highest speed database available, and in the no time you can get the sequences done. Now, comparison with other ribosomal databases. For example, if you have Silva, then you can have green genes. So these are some different databases where the first release is 1992. In Silva, it is the latest, 2007, and green gene is 2006. The host institute is Argonne early now, and Michigan State University now. Max Planck Institute by Silva has been kept on the Max Planck Institutes, and then green gene is UCSD, Lawrence Berkeley, and sequence type is 16S and 23S, bacterial, archaea, fungal, and LSU in this, and then in case of Silva is 16S and 18S SSU, that is 23S and 28S in LSU for all domains of bacteria, archaea, and eukarya. Whereas in green gene is 16S bacteria and archaea. So there's a variation in the databases, their mode of establishment and their execution and their database availability, their access and on. Different features also varies. Now, as far as update is concerned, regular latest 2023 was updated, and regular again Silva is also regular. Then green gene is basically 2013 onward. For last 10 years, more than 10 years, it has not been seen to be updated. So definitely RDP and Silva must be better than green gene. As far as the size is concerned, it is a moderate curated, the RDP, whereas the Silva is basically very large, billions of, because it has both prokaryotes and eukaryotes with multiple rRNA sequencing, both 16S and 18S, and then you have 23S and 28S in case of prokaryotes and eukaryotes, and both the ribosomal RNA sequences were taken into consideration. Then in case of green gene, it's a medium sort of thing, and then tools are basically classifier, aligners, probe, match are present in the case of RDP. Then database plus alignment is present in SILVA, and statistical or static taxonomy present, but there's no tools are available in the green gene. Now the current use is still popular classifier, Ribosomal Database Project, and SILVA is widely used in though it's better than in certain cases with the RDP, but still RDP is more popular. It's rarely used, outdated, because the database is not being used, not being updated further. Now, what is the application of Ribosomal Database Project? The major application of Ribosomal Database Project is the identification of the microbial species. To look into the diversity, to look into the ecological and diversity of these microorganisms. Taxonomic profiling of microbial communities like soil, water, gut, microbiota, extreme environment, right? So microbial diversity, whether it belongs from gut microbiome, from vaginal microbiome, or from oral microbiome, or from some environmental microbiome, soil, water, sewage, hospital, clinical, can be analyzed by this database. So you pick up the species, isolate and get a sequence, 16S RNA, and then you identify from which species it belongs. So they can give you the very clear-cut picture about the exact species in which the sequence is being belong. The environmental industries and microbiome. In case of environmental industrial microbiology, the monitoring microbes is bioremediation, right? What does the bioremediation means? Bioremediation means like you can identify certain microorganisms which are involved in bioremediation, pollution control, treatment of water, right? Suppose if you look into the sewage water, that is in many places there's a sewage water plant treatment system. So you can have water treatment plant, sewage water treatment plant, where there's an influx of several microbial population is supposed to be in added, and this population has to be identified in terms of like species level. So if you don't have a metagenomics analysis, or even the metagenomic analysis sequences can be also used to be used to understand by Ribosomal Database Project. Now, having multiple species identified from diversified microbial flora, which varies or which has a lot of diversity in their ecology, in their physiology, in their metabolic responses, in their texture, morphology, and genetics, can be easily identified, can be easily picked from this databases by just providing the sequences. So what is being done? Only you pick up the sample, water sample, during fermentation, what kind of a species. For example, if you are wine productions or any fermented food product where you are looking into what kind of microorganisms are present. So this is also used when you are trying to purify the best quality strain for production of fermented food. Now, once you identified these fermented food of a good quality, you isolate the strain, sequence it, find the specific species, right? And even you can also go for fingerprinting of that strain. As yesterday's my lecture on the molecular typing and molecular typing of the different strains of the species can be identified. In that case, you can also associate certain markers, specific markers, and these markers can help you out pick up the strains just by looking at the marker. Do the PCR and get the marker. So here we are talking about not the marker, but we are talking about more of like specific species from the different ecological and different applications, right? Now, identification of beneficial strain. I've just talked about probiotic strain, plant growth, promoting bacteria. So all these things can be done through this databases. Now, databases will help you to provide you the best possible information available based on the sequence that you got it from that particular situation or particular setting. Now, clinical microbiology, I missed it earlier. So it is the identification of pathogen. Now, suppose if you talk about the pandemic, say for example, during COVID-19, there's a lot of mycorrhizal strains of fungal species were identified. Black fungus, what you call. This black fungus, when identified, it has to be identified. Sorry, has to be identified through the sequences. Now, once you got this fungal from that site of infection as clinical setting from patient or from from surface of the incubators or surface of any machines or setting in the hospital or hospital tap water or from hospital floor or cords or any incubators or any stuff in the hospital, then you get the sequence it. Now, once you get a sequence, 16S RNA or 18S RNA in case of a fungus, then you compare this to the database. So now, without having that database, without having that ribosomal database project, ribosome database, you are not able to find the species exactly what is being found. You may get sometimes you may get some confusion in the identification that it is whether it is a bacteria or a species after they got a sequence. So just go through the 16S RNA. You find if you got some strain through 16S RNA, you can say, okay, this is the bacterial prokaryotic sequences. If not, it may be eukaryotic sequences, because it is not possible that you may not get you will not get the 16S sequences analyzed through the RDP. Yes. But in case if you have that sequence is not available in the databases, then you will not find. So in that case, you have to first, before check whether it's a new species or the old species, what you have to do is you have to go with this 18S rRNA sequencing. Now, once an 18S rRNA sequencing is being sequenced, then you can take this 18S RNA sequences, check through the databases, and you can say, okay, this is the fungus species. Now, in case if you have a situation where the 16S RNA sequences and 18S RNA sequences both are not matching in the RDP, it means you miss that strain from that databases. It means either that strain sequence is not added or not present in that databases. It means databases is not been updated. Number one, or that is a new sequences. Obviously, it is a new sequences which has not been updated in the databases. So for that, you have to contact to the authorities, to the person concerned, to the several authorized organizations who can give you the name of the new strain or new species, and the sequence has to be submitted again this RDP project to update it, and then your sequence will be updated as by your name or by some names which has been discovered by you as the first strain. So this is how it has been working to update the databases to get the best possible answers of the sequences you got through these settings. So this is one of the most important application in clinical setting, and that too also help you to identify the epidemiological aspects of these clinical strains, because that give you idea about whether the strain is epidemic or non-epidemic. Okay. Now then come to the phylogeny evolution. What is a phylogeny evolution means? Phylogeny evolution means how the strain varies on the time scale during the evolution, and what are the changes, and does these changes does this change make the strain some new variants or some new type or some new even species. It depends on what sort of variation and mutation might take place. So phylogenetic analysis and evolutionary significance of this databases also, because it can give you the clear idea about the species evolved over a period of time based on the conserved, and the variable regions, as we have discussed in the case of 16S rRNA, there's a lot of variable regions. So these changes taking place in variable regions, but not in the conserved region. So remember, these changes are not present in the conserved region. So if the changes taking place in the conserved region, then probably you cannot analyze it as a 16S RNA to identify the new sequences with the universal primers. Now then, bioinformatics methods in development are there. Then you can have RDP classifier widely integrated into the pipelines. So you can have a different pipelines too. So this is all part of the manufacturing part of the databases, how databases have been developed in order to provide the best possible results. Well, there are some limitations also, and these limitations are basically not much, but I think we have a small sequence coverage, data sizes, then because for the same reason, for example, if you have missed some sequences, then probably you may not identify the new sequences. The risk of missing rare novel taxa, as I have given an example like Serratia lepagi. See, Serratia lepagi has rare strains. If the database is not available, this sequence is not available in the databases, then probably you miss that sequences out of it unless you confirm its presence in the databases. So the bottom line is that you have to keep updating the database. The limitation is that if you are not going to update the database will not provide you the best possible answer of your questions. Now, 16S rRNA gene cannot always resolve down to species or strain level. So, if you are trying to find out the strain identification, for example, E. coli has 6,000 different strains. Now, these 6,000 different strains are within the species of E. coli, cannot be differentiated by 16S RNA, noted down, because as I've told you earlier that for that you need to have a fingerprinting method like RDDP, then you can have some RFLP, TRFLP, and so on. Closely related taxa, for example, E. coli versus Shigella, often indistinguishable. It's very difficult to distinguish sometimes because of the lot of variable lesions which are overlapped, and because of this overlapping of the variable lesions, it sometimes differentiate between E. coli and Shigella might be difficult. The strong focus on the prokaryotes, limited support for the eukaryotes, because in eukaryotic databases are limited. So it is very difficult to get the eukaryotic comprehensive data from this RDP, which probably need to be updated. Accuracy relies heavily on the quality and recently training set. So what are the training sets we have used in the databases to compare what is the purity, what is the quality, what is the percent homology, right? If it is 99.9% or 99.8% or even 99%, or if it is less than 99%, then probably it is very, very difficult to certain that, okay, this species belong to this sequence belong to this species. These are some limitations. New organisms may remain unclassified until future update, because if you don't have updated in databases, then you cannot classify that new organisms. Now, SILVA comprehensive and GTDB genomic based taxonomy are often preferred in large scale projects. So this is how we can make use of these databases over the RDP databases. So these are some references where you can refer it from these books, and I think this is all done. And with this, I close this lecture today. Thank you very much.